All right, so here we're going to pick up with part two of our general sensation lecture, talking about some of the structural components and structural features of different types of sensory receptors, and then we'll go in and we'll look at some examples of specialized sensory receptors. So, some of our sensory receptors are considered unipolar neurons, and so we discussed unipolar neurons whenever we did chapter 10, I believe. And if you recall, unipolar neurons have a single long projection that kind of runs in either direction with respect to the cell body. So if we're looking here at this example here, we see a cell body, we see this slight projection out, and then we have this one long process that extends out in either direction. So this is referred to as a unipolar neuron. Most of our sensory receptors, or sensory neurons, as they're coming back into the central nervous system associated with that dorsal root and the dorsal root ganglion, these are generally unipolar neurons. And if you recall, that cell body is located within the dorsal root ganglion, that kind of swollen bulbous region that you can kind of see in this region here. So this is an example of the unipolar neuron with the cell body in the dorsal root ganglion. Okay. So we also have unipolar neurons that have encapsulated dendrites. So if you notice here, we have free nerve endings, right? So we have a free nerve ending at the end of this unipolar neuron. We also have a unipolar neuron, but this one is encapsulated. So it has these collagenous connective tissue rings, or these layers that sit over the top of the dendrite and serve to stimulate that dendrite during the presence of certain types of stimuli. So we'll see that these are generally involved with sensations of pressure along with different types of vibration. Okay, so we can also see sometimes we have sensory neurons that synapse directly onto a sensory cell. And so here we have, this would be something like an example whenever we have our smell or our sense of taste, and so gustation. We have a sensory cell here that's going to be receiving some type of stimulus, right? So whether that's chemicals or some type of mechanical stimulus, here's the sensory cell that's responding. And then this is going to relay the signal via the synapse onto this sensory neuron here. And this example is an example of a bipolar neuron. And we see bipolar neurons generally associated with some of our special senses. So things like smell or olfaction, along with our retina for vision. So when we do special senses, when we do that lecture, we'll see there's an entire layer of the retina that is known as the bipolar layer because it consists of primarily bipolar neurons. Okay, we also have neurons that have a peripheral process at the end, and these are similar to the ones we just talked about, but these have a kind of separate peripheral process at the end that's slightly different from these sensory cells. Okay, so all these neurons that we've discussed, so the unipolar and all these different types, mainly these sensory receptors, they're going to have varying degrees of sensitivity to the presence of a constant stimulus. So imagine you have some stimulus. It's pain, it's temperature, it's touch, it's pressure, it's light touch, whatever it may be. You have this stimulus that is constantly present. It is always there. Okay. Now it's not always in the sense of perpetual infinity. It's always there, but for the duration of a certain period of time, it's always there. And so the way the cell responds, it can be in one of two ways. It can quickly adapt. So in other words, we can notice, we can sense, and we can perceive this constant stimulus. And then after a second, we kind of become used to it. We don't really notice it as much. Or in the constant of that same constant stimulus, we can continually be aware, right? So as long as that stimulus is there, as long as we sense it, we're going to continue to perceive it. Okay, so if you think of something like this example here, you put a Band-Aid on your arm. You put some type of gauze or wrapping on your arm. Initially, when you put that Band-Aid on, you notice it, right? You notice it. There is this mechanical pressure applied by the Band-Aid onto our skin, and we'll see in a little bit that's going to stimulate different types of touch receptors, so different types of mechanoreceptors. After a while, though, you notice that you probably don't really pay attention to the Band-Aid being there. You probably don't notice it. You probably don't feel it. So what that means is that the sensory receptors that are responding to the mechanical pressure, the mechanical forces being applied by this Band-Aid, those receptors respond quickly. They respond rapidly and they adapt rapidly to that, so you don't really notice it while it's there. So you notice it as soon as it was put on. You don't notice it after a little while as it's on, but then you notice it again as you take it off, right? So typically these rapidly adapting sensory receptors respond to the onset of a stimulus and kind of the offset of it. So when it is applied and when it is taken away. Okay, so we can call these tonic and phasic receptors. Okay, so again, the idea behind adaptation is we're going to decrease our sensitivity over a certain period of time to the presence or in the presence of a constant stimulus. So the stimulus is always there. The stimulus is the same in both of these examples for tonic and phasic. But how our receptors respond and how many action potentials they generate, that is what's going to differ. Okay, so for our first example is tonic. So tonic are going to be slowly adapting or receptors that have limited adaptation. Okay, so in the presence of a constant stimulus, they're going to initiate an action potential, and they're going to continue to send action potentials, continue to send information back to the brain, back to the central nervous system, as long as that stimulus is there. Okay, so they adapt slowly or not at all. So this is things like head position receptors in our inner ear. If you tilt your head to the side, so if you're sitting upright and you tilt your head over to the right, you'll notice that your head is tilting over to the right until you stop tilting it over to the right. So until you bring it back up. Also, our pain receptors or our nociceptors. Pain, and this is generally a very good thing, our pain is slowly adapting, or it has no adaptation, meaning we continue to perceive the pain stimulus, that pain signal, as long as that stimulus is there, right? So this is, for pain, this is a very good thing. We want to be aware that there is a painful stimulus as long as it's there, because we want to then remove our body, or remove our hand, or whatever is involved. We want to remove it from that painful stimulus, right? So if we look over here at the right, if we look at this graph, the top segment here is showing the stimulus, the middle section is showing the initial receptor potential, and then this bottom section is showing the number of action potentials generated. Okay, so here we have the onset of a stimulus. So we have no stimulus, and then we onset the stimulus, and for the duration of this yellow bar here, this yellow segment, that is the continuation of the stimulus. So as long as we have the stimulus present, we generated a receptor potential. So a receptor potential, if you recall, is similar to like a graded potential that we talked about when we did excitable cells. Okay, we did the action potential. But a receptor potential is specifically for a sensory receptor. So we have an initial receptor potential, and we continue to show the generation of action potentials for the duration of the stimulus duration, right? So we have the stimulus lasting this long, we have our receptor potential, and then we have the action potentials being generated this whole time. So it is slowly adapting. So in the presence of this constant stimulus, we're going to continue to generate action potentials. So that means that we continually send information to the central nervous system, so we continually perceive it, right? So this idea with pain, as long as you keep poking yourself with a pin, with like something sharp, you're going to continue to notice that, right? You have the stimulus constantly there. You have the action potentials constantly being fired, okay? So if you know tonic receptors and you know that they're slowly adapting, then you know phasic receptors because they're exactly the same except they're completely opposite, right? So these are going to be rapidly adapting. They're going to respond as soon as the stimulus is initiated, but then they're going to kind of go silent. And then as soon as you remove the stimulus, they're going to respond again, okay? So they only respond to new stimuli, so the onset and the offset. The action potential stops if the stimulus, the frequency of the action potential stops if the stimulus is unchanged. So if you continually have that action potential, eventually you're going to stop generating action potentials. And this allows us to ignore information and stimuli that's kind of always there, right? So we don't want to always pay attention to every little thing that is happening. We want to filter out what is not necessary, and we want to focus on what is necessary, right? So an example here would be things like pressure receptors, okay? So baroreceptors. So you may notice like the example with the Band-Aid for a touch, or if somebody pushes on your arm, or if you're in a crowd, you may initially feel when somebody pushes up against you, but after a while, if they continue pushing up against you, you probably don't notice it. And then whenever they move and they relieve that pressure, then you notice it again. This is also true for thermoreceptors, and a really good example of this is something like if you put your hand in water, or when you take a shower, or when you walk into a new room and that room is a different temperature from the room you just came from, right? So if you think of the example of like showering or put your hand in some water, when you initially introduce your hand or your body into that water, right, you notice that immediate change, right? So the stimulus isn't changing. So like you turn the shower on and it's really hot water, that stimulus is going to be the same. That water is going to be hot for the duration of the shower, or that hot water, that cold water. As soon as you get in the shower, as soon as you put your hand in the water, you notice it, right, very quickly. So you have a receptor potential be generated very quickly, and you have a few action potentials be generated right down here. So you notice it pretty immediately, right? However, after you've left your hand in the water for a while, or you've been in the shower for a while, you probably don't notice the temperature all that much, right? You kind of get used to it. You adapt, right? You rapidly adapt. So the water temperature didn't change, right, of your shower or what you put your arm in. It's still the same, but your response to it or your perception of it has changed, right? So after a while, you do not generate any receptor potentials. So if you don't have any receptor potentials, there's no way you can have any action potentials until you say, let's say we take your arm out of the water or you turn off the shower and you notice another change in the temperature. So right here, this receptor potential, and you generate some more action potentials, right? And then you'd get used to the temperature of the water after you remove yourself from it, okay? So this is this idea between rapid adaptation or slow or limited adaptation. The stimulus length is the same, right? So in both of these, constant stimulus, but what changes from tonic to phasic is how our receptors respond, or how our receptors adapt, or how quickly they adapt, okay? Are we responding very slowly or not at all? Do we continue to generate action potentials, or are we adapting very, very quickly, very rapidly, and we only generate action potentials at the onset and the offset of it, okay? So hopefully this makes sense, and think of these examples as you're thinking about this, okay? So again, this idea of sensory adaptation is a loss of responsiveness. So there's a few graphs here that can also help you understand this. And these graphs are very similar to what we saw in this graph here. So what we're looking at here is a slowly adapting receptor. Okay, so slowly adapting, remember, is tonic. So what we have here is we have the onset of a stimulus, and it's slowly increasing. And so as it slowly increases, we slowly have more frequent action potentials. Okay, but what you notice for this one is as long as the stimulus is there, so along this kind of straight line, you continue to have action potentials, right? The only difference between this top one and this bottom one is how quickly you reach your peak stimulus. So here you gradually reach your peak stimulus, here you quickly reach your peak stimulus, right? If we look at this example, here we're now in a phasic. So you remember phasic, fast adaptation, rapid adaptation. Here we are slowly introducing more stimuli, so we generate more action potentials because we're increasing the stimulus. But once we reach peak stimulus, you notice we're a constant stimulus, so we're going to adapt rapidly, and you're not going to have any new responses. So for the duration of this action potential, this intensity, as long as it is constant and there, we're not going to have any action potentials. And you see the same thing for this example down here, except instead of gradually increasing intensity of the stimulus, we quickly increase it. But you see the same thing. As long as that stimulus is constant and steady and there, we do not generate any subsequent action potentials, right? And then this example is just an extreme example of phasic adaptation, okay? So depending on these sensory receptors that we discussed and how broadly distributed they are, we can have something known as a receptive field, okay, based on the size of a sensory unit. So a sensory unit is one neuron, one sensory neuron, and all of the sensory receptors that belong to that one neuron. So if we're looking at this example, let's look at this right one. Here's one neuron, right? So here's an axon of a neuron. And then here are all of the receptors in the skin, all the receptors associated with this one neuron, okay? You can also look at these to the left. You can see one axon of one neuron, and you can see its receptors. You can see another axon of another neuron, and you can see all of its receptors. So the size of a sensory unit is known, and the amount of tissue it covers, the amount of skin it covers in this example, is called a receptive field. All right, and so there is a test that you can do very easily at home, and it is called 2-point discrimination. Okay, so a 2-point discrimination test. And so the idea behind this 2-point discrimination test is it plays off and has to do with the information of a sensory unit and a receptive field. So the larger the receptive field, okay, so the greater this is, depending on the size of the sensory unit, the greater the receptive field, the less likely you are to be able to differentiate between 1 point and 2 points on a certain part of your body. So if you have a section of skin that is very large, or very large in terms of one sensory unit supplies a lot of that section of skin, and you put 2 pieces or 2 points, so it could be the ends of a staple or end of a paper clip or something like this. You put 2 points on that section of skin, and both of those points are within the same receptive field. If both of those are within the same receptive field, you are more than likely going to only feel as if there was 1 point touching you, even though we can see here, even though there's 2, right? It's touching here, it's touching here, but you only have 1 feeling. You only perceive 1. This is because both points are within 1 receptive field, right? So it's transmitted along 1 axon, so you perceive it as just 1 touch, 1 point. However, if you have smaller sensory units, so you have a smaller receptive field, and you have the same thing happen, you have 2 points come down and touch your skin. If both of those points are stimulating receptors of a different sensory unit within a different receptive field, then you're more than likely going to perceive that as 2 different points, right? So here on this example on the left, we have 1 point here, stimulates this sensory unit with this receptive field, and this one here on the right, this sensory unit with this receptive field. So you have stimulation of 2 different sensory units because you had 2 different receptive fields. So you're likely to perceive that as 2 separate points, and that's exactly what you see here. So this is you can test this by touching, using 2 points and touching the skin at the same time along different parts of the body. So you could try the upper arm, you could try the forearm, you could try the fingers. Now, the relationship between this is inverse. So if the receptive field is very, very large, then you have a smaller chance of being able to precisely locate that stimulus or being able to tell that it was 2 points versus 1 point. If it is a very small receptive field, like we see on the left, then you have a greater likelihood of being able to tell that there were 2 points there because the receptive fields are smaller, so you're more than likely going to stimulate 2 different receptive fields, so you're going to feel it as 2 different points. Okay, so hopefully this makes sense. This graph here is showing how you can test this yourself. So you could do it on the tongue, you could do it on the finger, you could do it on the shoulder, you could do it on the forearm, wherever. Okay, so in this example here, you could use a paper clip if you don't have one of these actual instruments. Okay, and again, this is called 2-point discrimination, or they're calling this the 2-point touch threshold test. Okay. So we're going to stop here, and we will pick up with the next part, talking about specific types of sensory mechanoreceptors.